Here is 3 o'clock. Thank you all for joining us. We'll probably have a few more hop on here. Welcome to the SciCafe in August. SciCafes are so that we can, it's a platform for us to share science that's being conducted in Apalachicola Bay and other areas in an informal, casual setting, bringing together residents and scientists to explore different topics. Will be about 35 to 45, 50 minutes, and we'll have plenty of time for questions. Today we have Jason Garwood with us, and Jason graduated from the Coastal Carolina University. He's actually been an employee of the Research Reserve for 13 years. And he currently serves as the coordinator for the research section. Today we're going to talk about red tides and other harmful algal blooms, and it's understanding HABs, which are harmful algal blooms in Apalachicola Estuary. Last fall, as you know, we had a brush with red tide, and everybody was quite concerned, as should be. Although they're very common off the Gulf Coast further south, we do not tend to get them here. So Jay monitored that event, and he's going to talk to us about harmful algal blooms, or HABs, to shorten it, and answer our questions at the end. So Jay, take it away.
All righty, can you guys hear me okay?
Yes, we can hear you very well.
All right, greetings everyone, and thanks for taking the time and the interest to stop by and listen to me speak this afternoon. As Nita said, my name is Jason Garwood, and I'm the research coordinator at the Apalachicola NER. Before we get started on the main topic for today, I wanted to take a little bit of time to talk about who I am and what I do for the reserve. One of the things that a lot of folks don't realize is that NER is more than a facility for education and training. It's also home to a fully functional research program and laboratory. And it's part of my job is to make the day-to-day activities run smoothly. So obviously, the first and most importantly, the main task of my section is research. My department's involved in a wide-ranging variety of different research topics, all of which are centered around the Apalachicola Estuary and the associated river and floodplain. Much of the work that we do is centered around our system-wide monitoring program, which is a federally mandated requirement for all 30 reserves that exist around the coastal United States. And we do a lot of research here at the reserve. As research coordinator, it's my job, obviously, to coordinate the research, but in order for me to do that, I'm not able to do that without having a team of scientists and a laboratory. As part of our program, we do things like data collection, we conduct data quality control, we store and manage data as well so that it can eventually be analyzed. And we also collaborate with outside researchers from outside of the reserve. For example, we work with universities, more specifically, professors and their students, as well as other local, state, and federal agencies. And then finally, another aspect of what we do is we communicate our data and findings to the public and also interested stakeholders, like I'm doing for you folks here this afternoon. So just talk a little bit about what we do here. One of the most important programs here, research programs here at the NER, is what we call the System-Wide Monitoring Program, or refer to it as SWAMP. SWAMP is a nationally mandated NER program. It consists of 3 main components, that being water quality, nutrients, and meteorology. And these 3 programs are run in the same fashion no matter which other reserve you're at. And what this does is it enables us to look at baseline conditions as well as any deviations from baseline that might occur within a site. And because these programs are identical, no matter where you go, we can also look at changes in estuarine conditions on the national level as well. Currently here at ANER, we have data going back all the way to the early mid-90s, and some of these have been collected at 15-minute intervals, 24 hours a day, 365 days a year. In addition to our swamp program, we have what we call a biological monitoring program and also a climate change monitoring program. For biological monitoring, we conduct research on fisheries populations, plankton populations, as well as marine and marsh plant populations. And then finally, for our climate initiatives, we monitor changes in temperatures, sea level, and also whether or not our local marshes are able to keep up with the changes that we're seeing with our local sea levels. So, I've only just touched on the surface of what my team and I do. So if there's anybody on here that has specific questions about our research, please feel free to reach out to me and we can discuss whatever you would like in much greater detail. But without further ado, I want to move on to today's topic, which is about HABs. Just before we start, I'd like to put forth a bit of a disclaimer that I'm not in any way a HAB expert. My staff and I collect HAB samples on the regular basis during our swamp nutrient sampling. However, those samples are sent off to the FWC lab in St. Pete, and those are processed and analyzed there. So we don't store any of the HAB data here in-house. And so the small amount of data that I'm going to be presenting today, I essentially got through a public records request through the FWC. And so what I'm going to tell you about HABs is just sort of my own personal but well-informed opinion on what we've seen here in our system. And then I'm just going to relay this information to you guys because of concern over a lack of public understanding of what these things really are. So if you're looking for, an official statement about HABs, then I would highly encourage that you reach out to the FWC. So for the rest of the talk, I'll be talking about four main topics. First, that is HABs, the who, the what, the where, the why, and the how. And then next, I will move into HABs occurring in Apalachicola Bay. And then for this case, I'm going to be focusing mostly on red tides for the day. And then I'm also going to talk a little bit about the Florida Gulf Coast red tide event that occurred last year. And then I want to talk some more about how we can mediate or mitigate the effects of these events. And so to lead into that, I was going to show this photo here of one of my staff, Ethan, who is in a hazmat suit. And he's wearing this suit as part of our training that we have to take each year to be able to respond to potential hazardous spills in the bay. But I just wanted to clarify that some of the fears that surround HABs, and that although they can be potentially dangerous, a little bit of education can go a long way to protect you and your loved ones from harm related to these events. And even more benefit could be done if we could work together to limit the occurrence of these events in the first place. All right, so first of all, what are HABs? The most generalized term, HABs are just an acronym to describe a harmful algal bloom. So what is a harmful algal bloom? Harmful algal blooms basically occur when colonies of algae are out of control and produce toxic or harmful effects on people, fish, shellfish, marine mammals, or birds. In general, HABs are caused when algae, which are simple plants that live in the sea and freshwater, start to grow out of control. And we know about these because of their harmful effects, which have the potential to cause human illness, which, although rare, could be debilitating or even fatal. So before we can talk a little bit more about HABs, I wanted to first get folks to understand, what algae are in the first place. And so in the most simplest terms, algae are plants. They're simple plants that live in the sea and the fresh water. And if you look at the diagram on the top right here, we can see the differences between the soluble plant versus the soluble animal. And one of the biggest differences in the two is that both their structure and function. For example, animal cells respire, they metabolize sugar, utilize oxygen, and produce carbon dioxide, as opposed to plants which utilize light energy to produce carbohydrates, metabolizing carbon dioxide and producing oxygen. Plants can be unicellular or multicellular. However, traditional plants are always multicellular. And unlike algae, they have true tissues. So going into more detail, if we look at the diagram on the bottom right, we can see that algae can be unicellular, they can be filamentous, or what they call thallus in their structure, whereas plants always have roots connected to a trunk that extends to the leaves. So some algae can actually appear to be a multicellular plant. For example, sea lettuces that comprise the genus Ulva, which are a group of edible green macroalgae that's widely distributed along the coasts of the world's oceans. So we also have different types of algae. So there's macroalgae and there's microalgae. And just a few examples of macroalgae, and one of those would be something called red algae. A common red macroalgae is Gracilaria, which is a genus of notable for its economic importance as an agrophyte, as well as its use as for food for humans, as well as different species of shellfish. We also see brown algae. For example, kelp are a common large brown algae that live in cool, relatively shallow waters close to shore. And then again, I mentioned green algae, ulva, which are commonly used as sea vegetables and salads, and it's also served cooked in things like soups and along with meats and fish. So next, when it comes to HABs, the type of algae we're referring to in this case would be microalgae. And in the marine environment, these are commonly called phytoplankton, which are mostly microscopic, single-celled photosynthetic organisms that live suspended in the water. Like all plants, they metabolize CO2 to make carbohydrates using light energy, and they release oxygen. They're important in that they're known as the primary producers of our oceans. And so these organisms are the ones that form the base of our estuarine and marine food chains. And they have to live near the surface where there's enough sunlight that can penetrate to power photosynthesis, which in this area is what is referred to as the euphotic zone, which ends up being the top 600 to 900 feet within our world's oceans. And so the photos on the bottom here are examples of phytoplankton that we collect as part of our zooplankton research that we do. And so we see these here in Apalachicola Estuary. And these are very tiny, but they can be seen with the naked eye upon really close inspection of the water. Okay, so for today's discussion, the microalgae comprise two different kinds of organisms. The larger category would include the single-celled algae known as protists, as well as other phytoplankton that we're talking about today. There is another group of photosynthetic bacteria known as cyanobacteria, which typically cause the blue-green HABs that we see in freshwater. The cyanobacteria are among the oldest known living organisms on Earth, and they're understood to be the origin of the photosynthetic organelles in our modern plant cells, essentially known as chloroplasts. So basically, some of these ancient species are thought to have evolved from the cellular into the cellular organs of our modern day plants. Some cyanobacteria, again, can cause HABs, but for today's talk, we're not going to be spending much time on this group. All right, so in detail, phytoplankton are extremely important. And they're important because one of the reasons because they generate about half the atmosphere's oxygen. So it's not just our trees and our crops and all the other plants that we see on land. It's also the plankton that are in our seas. And because of this, they actually form the base of virtually every aquatic and marine food web. So in short, they make most other ocean life possible. And one of the ways they do that is they transform inorganic carbon in the atmosphere and in seawater, which is less bioavailable, into these organic compounds, which are then more bioavailable. And that makes them an essential part of our carbon cycle. And they do this by removing CO2 from the atmosphere when they die. They rebury back into the earth in other forms of carbon, which that makes them extremely important actors in the Earth's climate regulatory system. So one of the most important take-home messages that I want folks to receive is that most plankton or algal blooms are essentially a natural and necessary function in the estuarine and marine ecosystem. And I was just going to provide an example or two. One of those here is the Peruvian anchovy fishery. In general, these deep sea currents tend to be very rich in nutrients, and this is mainly because it's due to the decomposition of plants and animals that were present when the body of water was located at the surface of the ocean. This deep ocean water flows along the bottom of the ocean until it hits the continent of South America and then is consequently forced upward to the surface. And is where these surface equatorial currents are able to then converge, and they essentially concentrate these highly nutrient-rich waters where there's very abundant light, thus fueling these highly productive areas of Pacific Ocean. And it's these quote-unquote blooms of phytoplankton that fuel the rest of the food web, including anchovies, which feed on the zooplankton that are fed by the phytoplankton. Thus, anchovies are then food for other fishes and humans. And to just kind of bring this closer to home, we see similar nutrient plant productivity processes occurring right here in our Apalachicola Estuary. So here in Apalachicola Bay, phytoplankton or this algal growth occurs every single day, and it's necessary. A large part of my team's research activities revolve around monitoring the variation in these processes. For example, our water quality and our nutrient and primary production, as well as our plankton monitoring, are designed specifically to measure the spatial and temporal variation within our estuary. And the map here on the left shows some of our sampling locations that we visit, as well as the center images that show the type of net we use to collect these organisms within our system. So one of the ways that we can do this here in our system is by monitoring a chemical called chlorophyll, which is a chemical that's directly related to the abundance of phytoplankton that are undergoing this process of photosynthesis. And what we can see is that the area of our system that has a tendency to see the greatest amount of this production happens to be up in the East Bay region, which in fact is closest to the Apalachicola River, which obviously is a major source of nutrients to our system. However, if you were to look at the chart on the right, the time of year that we tend to see this great amount of productivity tends to be in the fall, which is a time of year that, although river flow has a tendency to be lower or less, or in terms of less nutrient input, the waters in the bay also tend to be clearer, which allows this deeper light penetration. And so the point that I wanted to drive home here is that there's a very delicate balance between food and energy here in the natural environment. And when that balance is removed, that's when we can begin to see problems. And that's what then leads us to question why or how do these blooms change to go from beneficial to become harmful. So sometimes these blooms have a negative effect or impact on humans or other animals, and so they're harmful. But why? And it's because sometimes these blooms become so dense that they can deplete the oxygen in the water, and it can cause fish and shellfish to essentially suffocate. That's not all. If the right species of algae happen to be present at the time when these conditions are right, their proliferation can not only cause a depletion of oxygen, but they can also produce toxins that can cause illness or death among humans and other organisms. These are the HABs that can cause significant economic loss in our seafood industries, as well as in our tourist communities. So what is it that causes these HABs then? And the simple answer is eutrophication, which is defined as excessive richness of nutrients in a body of water, frequently as a result from runoff from the land. These excessive nutrients can cause an overgrowth of plant life and death of animal life from the lack of oxygen. However, I have an asterisk on this because I would say there's 3 ways that this can occur. Obviously, the first would be lack of oxygen. Another way would be toxicity. And then again, there could be any combination between the 2. So it's at this point, hopefully, some of the folks in the audience are questioning, how would this be the case? On several occasions, I've said these are plants, and we all know plants produce oxygen when they photosynthesize. So if this is the case, then how are they producing oxygen and depleting oxygen, causing suffocation all at the same time? And our answer to that is bacteria and decomposition. So as nutrients become depleted from the production of these algae, then light becomes limited because of a decrease in the clarity of the water body. These algae then start to die. And as they die, they begin to undergo decomposition via bacterial activity. The bacteria use up the oxygen in the water column through respiration. And so, often regarded as vectors of disease, most bacteria are harmless. In fact, they're integral to our ecosystems in that they break down organics and make nutrients more bioavailable. They're also extremely abundant. So just in a single drop of water, there can be over 100 species of bacteria. And like humans, bacteria respire. So they consume oxygen and they expel carbon dioxide. And what this does is it basically puts the system into this feedback loop where we get excess nutrients, we see excess plankton, we get excess bacteria, and thus we get insufficient oxygen. So I want to talk a little bit about toxicity, and I want to do this by talking about just a few species that are common in Florida. Some of these species can produce toxins, and under normal circumstances, these are either not present in the water column or they're in a low enough abundance that their toxins don't cause any harmful effects on the environment. However, when they do, their effects can be substantial. And I've listed just a couple of the more common offenders here, along with their effects on people. So the first one is the red tide caused by Acronia brevis. It's known for the red pigment that they produce when undergoing overgrowth. And when ingested, these red tides are known to cause what's referred to as neurotoxic shellfish poisoning. And in this case, the toxin that they produce is called brevetoxin, and it can cause gastrointestinal as well as neurological dysfunction. However, a common side effect of these, as some of you might know, is respiratory and skin irritation. And fortunately, they're rarely fatal to humans. And another type is pseudonychia, which is another type of plankton that we can see. It's able to bioaccumulate enough in food to cause what's known as amnesic shellfish poisoning. And in this case, domoic acid is the main toxin for the species. And like like these red tides, it can cause gastrointestinal as well as neurological disorders. And common symptoms would include dizziness, headache, seizures, disorientation, and respiratory dysfunction. And if this occurs with the wrong person, it could potentially be fatal. And then another one is one that's called Gambier discus. It's another one that's found in Florida waters. Humans are affected by this particular algae by the ingestion of toxified finfish. So this would be your snapper, your grouper, your jacks, etc. And Gambier discus produces a toxin called ciguatoxin, and the poisoning is called Ciguatera fish poisoning. Symptoms for this particular algae include gastrointestinal as well as neurological and cardiovascular dysfunction. And it has been known to kill people. I actually have a friend who has actually had this, has a really cool near-death experience from eating barracuda. So it's a real threat if you get this stuff in the wrong concentrations. Okay, so let's talk a little bit more about red tides. These HABs do occur here in our bay, and out of interest in putting this talk together, I got curious about what the Carenia brevis abundances have looked like over the past few years. So what I did was I requested some available data for the species for the Apalachicola Bay from 2018 to 2021. And these are samples that are collected by my staff here during a regular nutrient sampling, as well as the Department of Agriculture and Consumer Services and the FWC. And I believe the product preserves also sample these as well. It's FWC that manages the database for these. And what we can see here is that there's definite overgrowth of Carenia brevis in both the fall of 2018 with this huge peak during the fall, coinciding with that big red tide event that we had last year. Because of the scale of this event, it masked some of these smaller peaks that we had at other points during the time frame. And what it appeared to me was that there's maybe a tendency for some seasonal peaks of this species, more specifically during May and November of each year. And so I think it'd be interesting to go back further in time, maybe follow this into the future as well, and see whether or not this has been a recent or an intermittent phenomenon, that something that's been occurring, and will continue to happen into the future. All right, so I want to talk a little bit more about HABs here in the Apalachicola Bay. Let's look a little bit closer into past red tides, more specifically the way they tend to be associated with tropical systems. For example, in 2005, Hurricane Dennis was a Category 3 storm that came to shore around the Florida-Alabama border. There's a significant storm surge from this event, as well as rainfall, which allowed for plenty of runoff and thus plenty of nutrients, leading to local eutrophication. And what we saw afterwards was red tide effects that went into September of that year. And the photos along the bottom of this slide are of Little St. George Island, and they show the dead fishes washing to shore, as well as on the bottom right, we can see a dead cottonmouth snake that had most likely eaten the toxic fish and then become fatally poisoned. Another example would be the hurricane that we saw in 2018 in October for Hurricane Michael, which was a Category 5 storm that came to shore between St. Andrews and St. Joe Bays. Our swamp meteorology and water quality instruments were able to capture the conditions as the storm came to shore and subsequently moved out of the area. The data I have here, shown here, are from our station in the Little St. Mark's distributary of the Apalachicola River. And the top center graph shows depth and salinity at the station, which we can use as a proxy to assess the timing of the peak of that storm surge for the event. And the chart below shows daily change in oxygen dissolved in the water for that month of October 2018. And what we see was that there were 2 drops in dissolved oxygen coinciding with that event. First, a drop occurred on the 10th, the day of the storm, after which there was a slight increase, followed by another dip in very low oxygen that persisted for the remainder of the month. And what I believe we're seeing here are combined effects of eutrophication from runoff from water from the land to the estuary. And then I also believe the second would be that drop in oxygen, which persisted about a month, occurred because of a combination of a sewage breach that occurred upstream, as well as a small red tide event. For those of you that were here, you might recall that during this time we saw some significant fish kills in the area, which you might associate with very low oxygen levels in the water. And then the chart here on the right shows a peak in Carenia brevis in the bay of around 3,000 cells per liter, coinciding with the passing of the storm. Note that the data show Carenia brevis were already present in the system in September. However, it's likely that the passing of the storm provided the nutrients needed to cause that bloom. Right, so I want to talk a little bit about the 2021 red tide event that we saw. Once again, I just wanted to preface this discussion that this is just my interpretation of the event. It's not the state's official take on the incident. But like this event again was caused by Carenia brevis, which again is a microscopic single-celled dinoflagellate. It has 2 flagella, or tails, that allows movement against gravity towards light. And the species is common to the Gulf of Mexico. And the optimum environment tends to be a temperature range of around 22 to 28 degrees, which coincides to 72 to 82 degrees Fahrenheit. And they tend to like a salinity of around 25 to 45 practical salinity units. This species can metabolize both organic and inorganic nutrients, which makes it very good at utilizing nutrients from runoff. And they're known for their red pigment that occurs when in high abundance, and also their ability to produce the brevetoxin that's harmful in these high concentrations. And FWC first noted the first positive samples in these significant concentrations near the Charlotte Harbor area in the spring. Just as a note, not all dinoflagellates cause these negative impacts. So the image on the left is a dinoflagellate of the genus Serratium. It's from samples we collect here in the bay versus K. brevis on the right, which is much less common, but can cause the red tides we see here. Certain species of Serratium can cause blooms, but they won't be the focus of our talk today. So as I said, the first known positive samples of K. brevis occurred around Southwest Florida in March of 2021. And by July, this species had expanded up to the central Florida Gulf Coast around the Tampa Bay region. And then by September of that year, we were seeing hits as far west as Pensacola. And so it appears as though this bloom spread from the southeast to the northwest as the season went by. And just to kind of take a look at, a possible mechanism for this event, I went ahead and looked at local wind data collected from our system-wide monitoring program meteorological station located here in East Point. And in the chart on the left, I've plotted the mean compass directions from our anemometer, which measures both wind speed and direction. And what we see is that there's an overall tendency that year for these winds to come out of the southerly and southeasterly direction for the entire year. Therefore, if this pattern in fact persisted throughout the Gulf region, or the Florida Gulf region, then it's possible that the mechanism for the spread of the dinoflagellate throughout the season would have been because of these winds. And that would potentially explain why we saw this gradual spread over the spring, summer, and fall months. All right, so I want to talk a little bit about ways that we can, mediate or mitigate some of the impacts from these events. And one of the ways we can do that is by minimizing how they happen. And so one way that we can do that is by producing less eutrophication through environmentally friendly practices, such as farming methods that reduce nutrient shedding from land to adjacent waterways. We can clean up pet wastes in the area where runoff is prevalent. We can decrease the use of lawn fertilizers and also do practices that minimize the loss of those applied nutrients to our nearby waterways. Another way is that we can, provide effective wastewater treatment by minimizing breaches during power outages and storm events, and also proper and effective use and maintenance of our septic systems. Unfortunately, the reality of the situation is that it's likely that we'll see more of these events in the future, and they'll likely become more sustained and even maybe more severe. So I think it's important, if we can't prevent them, then we could at least lessen their impacts. And so, one of the ways we can do this is through safety. If you're in an area where that's known to have these types of blooms, always be sure to check for advisories when you're going to be recreating or drinking or doing any consuming food in those environments. Also, if you know that there's an active bloom occurring, just stay out of the water. And also, this is especially true for the young and our elderly and our immunocompromised folks. And of course, we should extend this same rules to our pets as well, keep those out of the water. And then, if you happen to be in an area where these blooms occur and you see that there could be one occurring, go ahead and report them, and so that state folks can start responding to their effects. And most importantly, if you find yourself having symptoms and you've been around one of these areas, don't ignore them. And if it starts to get to where it's worse than what you think you can handle, it's important to seek medical attention. Okay, so I just wanted to conclude and wrap up with just a few major points. One of those is that one of the most important take-home messages I want everybody to take is that most plankton or algal blooms are a natural and necessary function in our estuarine and marine ecosystem. Problems occur when these blooms or overgrowth from these microscopic plants start to go into overdrive. And these microscopic plants are normal and necessary, though. Some blooms can have harmful impacts on humans and other animals, and they're harmful in that they deplete oxygen and or produce toxins. And that we know that these are occurring typically because of something called eutrophication, which is this excessive richness of nutrients within a body of water. We also see that red tides do in fact occur here in our bay, and that there could be potentially seasonal patterns that we see here in our system. We've also showed that hurricanes can also produce these favorable HAB events. So we can sometimes expect, if we're having some of these tropical systems, we might see this happen. And then in 2021, it looks as though possibly the environmental conditions created this perfect condition for things like temperature, winds, currents, as well as the presence of the algae to spread throughout the Florida Gulf Coast. And then finally, we can mitigate by reducing our impacts on our environment and by protecting ourselves and our loved ones. And with that, I would like to conclude and take any questions.
Thank you so much, Jay. Alicia, you want to come on camera and do you have any questions? I had a quick question. Jay, I've heard of ciguatera mainly being down in the Caribbean. Is it made its way this far, or?
Well, no. So the example that I was referring to with a friend happened down in South Florida. It was down in the Keys somewhere, and yeah, he had eaten some barracuda and basically died on the table.
Really?
So yeah, ciguatera is not real common up here, but it's something to always to be careful with.
Yeah, probably better not eat barracuda.
Yeah, it's common in barracuda.
Yeah. Any other questions? To introduce Alicia Bruno. She is producing our meeting here and following up on the questions. So thank you, Alicia. And we thank everybody for coming today, and thank Jason for your talk. This will be recorded on our website at apalachicolareserve.com. You can listen to it. And then we have next month, September 22nd, we'll have Dr. Stephen Durham, who will be talking about what fossils can tell us about oysters in Apalachicola Bay. He is a paleontologist with the Department of Environmental Protection, and it should be a really interesting talk. And I see we do have a question.
Yes, we just got one. Jace, so Diane wants to know, cleaning up the dead fish that come to shore, is that a needed thing to do to shorten the red tide period?
Can you still hear me? Am I still on?
We can hear you.
Okay. So the question is cleaning up the fish, will that lessen the effects of, or the duration of the red tide? Okay, I don't think so. I think you're going to see these dead fish. And this is just my own personal opinion. I would expect that you'd see these fish continue to die either through, the toxic effects or, low oxygen effects until the proliferation of the algae have discontinued. So I don't think that. I'm not saying don't clean up these fish, but I don't think that it would lessen the duration of it.
Okay, thank you.
Okay, any more questions? And thank you, Jay, for giving us this great talk, and join us again September 22nd. Thanks so much.